Okay, good afternoon, everyone. I'm just watching the attendees join the meeting, so we'll just wait a little bit longer. It's just 1 minute before 3, so we'll just wait a little bit. Okay, good afternoon, everyone. Time is now 3 o'clock, so we'll start. Thank you for joining us today. This is our third virtual meeting. C is for chromatography on behalf of the Royal Society of Chemistry Atomic Spectroscopy Group. My name's Jackie Morton, and I've helped to organise this virtual meeting. Any errors that occur during this, of which there's been a few, are all solely down to my technical inability to do this, and so I apologise for that in advance. Today's meeting is entitled C is for Chromatography, and we have 4 speakers. We have Roberto Alvarez Fernandez Garcia, we have Christian Ward Dietrich, and we have Nancy Wana, and we have Elliot Hamilton. The format of the meeting will be that each presentation will run sequentially with each speaker speaking one after the other, and at the end we'll collectively answer any questions. Please feel free to type any questions in the chat on the side on the drop-down menu, and I will try to coordinate the responses at the end of the talks. If you wish to specify the presenter you wish to speak to and address the comments to, if I can't work it out, then please put the person's name, Roberto, Christian, Nancy or Elliot, at the start of the chat function. So the first speaker today is Dr. Roberto Alvarez Fernandez Garcia, and he's a postdoc researcher at the Mass Spectrometry and Biomedical Analysis Group of the University of Oviedo. He received his PhD from the same university in October 2020, and during his PhD he worked on the method development and the application of analytical strategies based on ICPMS for the analysis of small structures such as single particles and single cells. Roberto will talk to us today about his recent research on speciation analysis of nanomaterials using HPLC ICPMS strategies. Okay, so we'll just transfer to Roberto and we'll start the presentations. Hi, Roberto. I'll just work out the sharing. Okay. There we go. Okay, you just unmute and you're good to go.
Now, can you hear me now?
Yes, we can. All working fine, thank you.
Okay, and the slides also on? Okay.
Yes, all perfect.
So thank you, Jackie, for your kind introduction, and good afternoon, everyone. Thank you also for the opportunity of presenting the results of my research group about the speciation analysis of nanomaterials by using HPLC ICPMS strategy. First of all, nanomaterials are defined by the European Commission as material containing particles that can be unbound or forming aggregates or agglomerates, and where at least 50% of these particles have 1 or more external dimensions on the size range of 1 to 100 nanometers. The nanotechnology represents one of the key technologies nowadays, and the unique properties of the particles have increased their use in thousands of daily products and in a wide range of applications. However, the widespread use of these nanomaterials and their consequent emission to the environment might have an important impact on living organisms. In this context, not only the composition and the concentration of the nanomaterials affect their toxicity, We have to consider also other physicochemical properties such as their size, their shape, and also their surface coating. In addition, the degradation of the nanoparticles and the capability of releasing ionic species can influence the toxicity of the nanomaterials. So we need new analytical techniques that allow to distinguish between nanomaterials and their dissolved ionic species to understand better their behavior. In this context, nanoparticle analysis is a great challenge because their physicochemical characterization involves obtaining information about size, elemental composition, shape, agglomeration state, or surface modification. In addition, the quantitative analysis involves the determination of both the mass and number concentration of the particles. So in this regard, the inductively coupled plasma mass spectrometry, or ICPMS, it's a powerful technique for the characterization of metallic nanomaterials because it provides information on size, concentration of the particles, and of course, elemental composition. The ICPMS can be used in two different modes, by using the single particle ICPMS approach or by using hyphenated technique. Very briefly, the single particle ICPMS works as follows. It consists on introducing very diluted particle suspensions to the plasma so that each particle reaches the plasma individually and generates one ion cloud of ions that is detected in the ICPMS as very short transit signals. So to discriminate between individual particles, short dwell times are used. It is a simple method that provides mainly three types of information. The intensity of the signals depends on the mass of the element in the nanoparticle, and therefore on the size of the nanoparticle. The frequency of the signal depends on the nanoparticle number concentration. And finally, the background signal is proportional to the concentration of the ionic dissolved species. However, the single particle ICPMS size detection limits are limited by the sensitivity of the ICPMS for the monitor element. And in the case, for example, of gold or silver, is between 10 and 20 nanometers. So the other option is to use hyphenated techniques such as field flow fractionation or hydrodynamic chromatography. The field flow fractionation is a family of techniques that are based on a flat channel where a separation field is applied perpendicularly to the sample flow. Depending on the separation field, different variants of field flow fractionation can be used, and for nanomaterial analysis, the most used is the one that uses a liquid flow, and specifically the so-called asymmetrical field flow fractionation, or AFO. In this case, there are two opposing forces that drive the separation: the diffusion of the particles and the separation field. So the smaller particles, which have a greater diffusion, concentrate in the center of the channel that has a parabolic flow, and thus they elute faster from the channel. As you can see here, this technique allows the determination of the nanoparticle sizes, but has some disadvantages, like the special equipment that it requires. It is difficult to optimize. The recovery of the nanoparticles is matrix dependent, and in some cases is very low, and the ionic species are not detected. The other option is to use hydrodynamic chromatography, and in this case, the separation is based on the fact that the larger particles concentrate in the center of the parabolic flow and elute faster from the column. However, the chromatography resolution is very limited, as you can see here, for the analysis of 20 and 5 nanometer gold nanoparticles with similar retention times, and the ionic species have very low recovery. So what is clear is that there is a need of new analytical techniques that allow to obtain relevant information about the nanomaterials and their degradation process. And in my research group, we have been working in the last year on the development of ICPMS-based techniques like the HPLC ICPMS. So the first step consisted on the use of reverse phase C18 columns for the separation of citrate stabilized gold nanoparticles. The mobile phase consisted on a phosphate buffer and the addition of the surfactant SDS that was absolutely necessary to avoid the absorption of the nanoparticles with the packing material of the stationary phase. To optimize the separation, 5 and 20 nanometer gold nanoparticles were used, and two different columns which differ on the pore size. Under these conditions, the use of the 300 angstroms pore size column didn't allow to separate between the 5 and 20 nanometer gold nanoparticles. This is probably because the hydrodynamic size of these particles is too large to enter through the small pore size, and the particles elute very close to the dead volume. However, the use of the 1000 angstroms pore size column permitted the separation of the 20 and 5 nanometer particles, and a size exclusion mechanism was appreciated with the larger particles eluting first. In addition, a third fraction corresponding to low molecular ionic gold complexes was obtained as impurities of the nanoparticles, but that can be very interesting for further applications. Here in the slide, you can see the separation of 3 different size commercially available gold nanoparticles of 20, 10, and 5 nanometers that were separated but not completely resolved. However, due to the high reproducibility in the retention time, a reliable correlation between retention time and particle size was obtained and was also used for the characterization of self-synthesized gold nanoparticles. Here in the table, you can see the results for 2 samples that were measured by HPLC ICPMS and by transmission electron microscopy for comparison. And you can see that the results in both samples are in a very good agreement between the 2 techniques. The developed method was also applied to the analysis of real samples, such as extracts of human colorectal adenocarcinoma cells incubated with gold nanoparticles of 10 nanometers, and Wistar rat tissue samples after the intraperitoneal injection of the same particles. In the case of the cells, the nanoparticles were observed inside the cells by transmission electron microscopy, and the cell extracts were injected in the chromatographic system, obtaining only 1 peak with a retention time corresponding to the injected nanoparticle. However, in the case of the tissue samples, apart from the nanoparticle fraction, we obtain another one that corresponds to low molecular ionic gold species, maybe due to the degradation of the particles inside the animal and showing a different behavior of the particles between the studies in vitro and in vivo. The method was also used for the determination of silver nanoparticles, but in this case, to avoid the precipitation of silver ions with phosphate, an acetate buffer was used. Under these conditions, 20 and 10 nanometer silver nanoparticles were separated as you can see here in the slide. However, the main difference with the analysis of gold was that the ionic silver was totally retained in the gold. To overcome this important limitation, several complexation agents were used, and the best results in the recovery of ionic silver was obtained for the addition of 1 millimolar of sodium thiosulfate to the mobile phase. The addition of this complexation agent didn't affect the nanoparticles that maintain the retention time, as you can see here for the 10 nanometer silver nanoparticles. This method was also applied for the analysis of real samples, such as sport socks that contain silver nanoparticles due to their antibacterial properties, and these particles can be released to the environment. So to simulate a washing process, the samples were incubated during 2 hours at 20 degrees with a phosphate buffer that contains the detergent SDS. The solution was injected in the chromatographic system, and 3 different fractions were obtained. The first 2 fractions represent the dilution of silver nanoparticles, and by the retention time, the sizes were estimated to be between 20 and 40 nanometers for the first fraction and around 7 nanometers for the second one. In addition, a fraction corresponding to ionic silver was obtained from the cells. And another and the last application that I'm going to present today is the study of the cellular presence of biogenic selenium nanoparticles in yeast cells. Selenium is an essential trace element for humans that play critical roles in relevant metabolic processes, and in high doses, it has also been used in cancer prevention treatments. Thus, the dietary intake of selenium can be complemented with the use of selenium supplements. These supplements are usually based on selenized yeast that can accumulate up to 3,000 micrograms of selenium per gram. And these selenized yeast are produced by growing different strains of Saccharomyces in cultured media enriched with inorganic selenium. In this process, they change the sulfur atom in methionine by selenium in selenium methionine. In this context, the a minimum content of selenium methionine, as well as the absence of inorganic selenium, is typically verified in these products. However, the characterization of other selenium species in the selenized yeast, even at the ultra-trace levels, is a hot topic of research. And one of these species could be biogenic selenium nanoparticles that can be biosynthesized as a selenium metabolic detoxification process. It is a reduction in which the final elemental selenium is less bioavailable than the selenium oxygen ions, and thus it's less toxic for the cells. So to study the formation of these biogenic selenium nanoparticles, we have to lyse the yeast cell. The problem here is that the yeast cell, apart from the cell membrane, they have another thick layer that is the cell wall that provides an exceptional rigidity. So for this reason, time-consuming enzymatic digestions are carried out to lyse the cells. However, in our work, we propose the use of a mechanical lysis that consists of mixing the yeast suspension with glass beads, and then the mixture is subjected to an ultrasonic bath and processed at maximum speed during 5 minutes. The procedure is repeated twice to maximize the lysis efficiency, and finally the mixture is filtered to avoid the presence of glass beads and potential cell debris. So the mechanical lysis was applied to cell 1, that is a selenized yeast 35 reference material, and this material was analyzed by single particle, single cell ICPMS before and after the mechanical lysis. The single cell ICPMS approach shares the same principle as single particle, so a very diluted cell suspension was injected into the plasma, and the individual cells were detected by monitoring both phosphorus and selenium in triple quadruple oxygen mode. Phosphorus is a constitutive element of the cells that is present in all yeast cells as forming part of relevant biomolecules such as phospholipids in the membrane, for example. And selenium represents the uptake of this element during the yeast production. However, after the mechanical lysis, the absence of signals of phosphorus can be an indicator of the efficient cell lysis. The absence of selenium signals means that there are no selenium nanoparticles in the yeast cells, or that their size is under the size detection limit of the technique, that was 20 nanometers for selenium. So to investigate the presence of smaller selenium nanoparticles, we decided to apply our HPLC ICPMS method to the lysates of the yeast. Those are the results, in which you can see 2 peaks. The first peak indicates the presence of selenium nanoparticles with diameters between 4 and 6 nanometers. And these particles are under the detection limit of the single particle ICPMS. The second peak represents the presence of low molecular selenium species. In addition, to verify the presence of elemental selenium as a major component of the selenium nanoparticles, the separation was complemented with a derivatization with sodium sulfide. The sodium sulfide reacts selectively with elemental selenium, causing the disruption of the nanoparticles as well as the appearance of new chromatographic peaks that represents the formation of the product. Here you can see the results, and after the reaction with sulfide, the first peak disappears completely. And this is an indication of the presence of elemental selenium of the particles detected in this size range. In addition, you can see here the new peak that represents the formation of the selenium sulfide. So to obtain more evidence about the presence of these small nanoparticles, the fraction of the particles was collected and analyzed by high-resolution transmission electron microscopy. And here you can see the pictures that we obtained with particles with diameters between 4 and 7 nanometers, confirming the results of the HPLC ICPMS approach. So as conclusions, I would say that the development of the reverse phase HPLC ICPMS method, adding SDS to the mobile phase, allows the separation of nanoparticles according to their size and following a size exclusion mechanism. And this increases the variety of tools suitable for the analysis of metallic nanoparticles. This method also allows the determination of small nanoparticles as well as the corresponding ionic species, revealing the potential of the technique for biomedical and environmental applications. And last, the combination of complementary analytical techniques such as TEM, single particle, or HPLC ICPMS permits to characterize biogenic nanoparticles in a wider size range. I don't want to finish without saying thank you, of course, to my research group, the mass spectrometry and biomedical analysis group of the University of Oviedo, all the financial and instrumental support, and of course, all of you for your attention. Thank you.
Hi there, Roberto. That was fantastic. I've been literally writing notes faster than you were speaking. That was really, really interesting. Thank you. Okay, so we'll keep all the questions for the end and we will move on to our next presenter. Thank you. Let's just turn your camera off. It's time for me to introduce Christian. Our second speaker today is Dr. Christian Ward-Dietrich from the LGC in the UK. Christian obtained an MSc in Instrumental Analytical Science in 2003 for the Robert Gordon University in Aberdeen. And he stayed on in Aberdeen at the University of Aberdeen to obtain a PhD in Analytical Chemistry, looking at the accurate quantification of metalloproteins using isotope dilution. His work at LGC since 2010 has involved working with high accuracy inorganic analysis team. The focus of his work there is the application of isotope dilution techniques for high accuracy determination of elemental species in complex matrices with the aim of providing reference values, methodology and materials for analytical and clinical communities. Christian's talk today is entitled The Use of Chromatography for Elemental Speciation, Absolute Quantification of Selenoprotein P via selenium peptides. Okay, so I will change you over to the presenter. Thank you, Christian.
Okay, can everybody see it?
Yes, we
Excellent.
can.
Thank you very much for that kind introduction.
Okay, thanks, Christian.
Thank you for the kind introduction. Yes, I would like to present our work on selenoprotein P. Yeah, give you a quick introduction of our element speciation background, why we need selenium determination, then our experimental peptide selection, the optimization of our HPLC ICPMS setup, then the double species specific HPLC ICPMS approach, and a little bit about our results, the conclusion, outlook, and acknowledgements. Just a background to everybody, elemental speciation. Also, speciation refers to the distribution of a defined chemical species of an element in the system. Therefore, speciation analysis is the identification and quantification of different forms of an element in a given matrix. And for example, a couple of examples are, for example, the distribution between chromium-6 and chromium-3, inorganic arsenic and arsenobetaine, for example, or in our case, we're looking at selenomethionine, selenoproteins, or total selenium. And for speciation, you normally use to a high resolution separation techniques like HPLC, which you can also run in different modes. So you have a multidimensional use, and then you couple it to a sensitive element specific detector. In our case, it's the ICPMS. So why selenoprotein P? So it's the most abundant selenoprotein in plasma, and it's highly relevant for selenium transport and storage. It has a high relevance in human nutrition and is associated with disorders, for example, type 2 diabetes, and it also plays a role in cancer prevention and treatment. And selenoprotein P or selenoproteins in general are the active forms of selenium in the body, and there are selenium status biomarkers. Why do we need it, or what's the impact? So at the moment, there's currently a lack of reference materials and methods for the clinical community. And we also need to do research in the function of selenium, especially in terms of cancer. And clinical quantification methods available at the moment, we don't know about the accuracy, selectivity, and they are not validated. So protein quantification by organic MS was a very promising tool to deliver results traceable to these SI units. So our aim was then to develop and validation of the species-specific IDMS reference method for the accurate quantification of selenoprotein P via selenium-specific detection at the peptide level. What are the advantages of ICPMS for this inorganic selenoprotein analysis? So we've got a wide choice of reference material for total selenium, which can be used to characterize the selenoprotein peptides traceable to the SEI. Normally for the ICPMS, we don't need extensive cleanup and pre-concentration steps, so it's a better matrix tolerance and shorter preparation times. The signal in ICPMS generally doesn't suffer from signal suppression, and the similar response is given in different matrices for different selenium species. Because we've got an element-specific detection, we are only interested in a particular element. For example, in our case, it's selenium and peptides. So we don't really care whether there's any correlation with non-selenium containing species, because they don't actually contribute to our selenium signal. And the latest instrumentation in ICPMS offers good interference removal, isotope ratios at low signal-to-noise ratios, and improved LODs and LOQs, which are actually required for the selenium protein P quantification at low levels. And we also have got for selenium, we also got a choice of using several isotopes, so we're not limited to measure only one ratio. The experimental, so if you're looking at if you look at the right here, so at the left here, so that's the selenium amino acid sequence of selenium protein P, and it contains 10 selenium cysteine residues. And if we do an enzymatic digestion, we get 8 selenium peptides. So we initially chose 4 peptides and synthesized the enriched and the natural variants, and those were then fully characterized for the isotopic abundance, structure, efficiency, and using a combination of various techniques. However, there are several considerations you have to take into account when you want to couple ICPMS and HPLC for elemental detection. In ICPMS, we hate organic solvents because, especially at high concentrations, which is normally used if you do peptide or protein analysis, you normally work at really high concentration of acetonitrile methanol, but those are not very good because they can actually extinguish our plasma. Of course, with a mobile phase, you introduce possibly interfering ions and contamination into the plasma. The flow rate has to be matched to the ICPMS. So you've got a carbon effect on selenium, which means the response increases with the organic composition. The gradient program you have to take in consideration in general the runtime, because ICPMS is a little bit expensive to run. So you have to make sure that your sequences won't be too long, or your method won't be too long. And for example, that shows an example of what a high salt load can actually do to your interface cones. So it's completely blocked. So you have to be really careful to make sure you adjust your conditions to your ICPMS detector. It gives you a little bit, for those who are not familiar with IDMS or isotope dilution, so the approach is based actually on the measurement of isotope ratios in a sample plant and compare them against a well-characterized species calibration plant. So we've got our traceable primary standard, then we can characterize our secondary standards. In this case, the natural selenium protein P specific peptides. We've got our enriched peptides. We add them together. So we've got our calibration plant. We've got our serum, our sample plant. And then we can measure both solution without any further cleanup and pre-concentration, and we get results for the peptides. We need a couple of requirements to fulfill. For example, the matching has to be very good. The spike has to be used for the same plants. We have the full equilibration between spike and sample plant, and both have to be submitted to the entire procedure, possibly at the earliest stage. But if you do that, you've got SI traceability, you got the compensation of the carbon effect on selenium, you can compensate for any sample losses, and you minimize or eliminate mass bias and linearity effects. So you've got a lot of benefits out of it. A little bit of the experimental workflow. So first of all, as I said, we had to characterize our selenium peptide, both the natural and the enriched ones. We had to optimize triptych digestion procedure, alkylation. We have to optimize the HPLC separation, the ICPMS detection, and before we can actually do our final IDMS analysis of the serum reference materials. So the characterization of the peptides, initial analysis showed that we can only use 2 peptides of the 4. So we did the total selenium contents and the amount of selenium in the natural, and we did the abundance and the amount total and the amount of the selenium in our enriched peptides. That gives you a little bit the HPLC ICPMS analysis of those peptide standards. You can see, for example, here very clearly when we have non-alkylate peptides, we can see here we've got various peaks, and if we alkylate them, you got only one peak, which is good, so you can better attribute actually the total selenium concentration. And you can see also here the enriched one, so the blue one is 76 and 78. So in the natural one, the 78 is higher as the 76, having the natural abundance. And in our enriched one, we've got hardly any 78 left, but it's mainly 76, so which is good. So that's what we want actually. The problem with ICPMS, it's a powerful quantification method, but it can only tell the compound contains an element, in our case selenium, because all the structural information is lost. So what we can do, we can actually combine the ICPMS and organic mass spectrometry to get actually quantification via the ICPMS and identification via the organic mass spectrometry, and to actually minimize falsely assignment of signals and peaks. So that gives, for example, a signal. So that's the top one. The top chroma is the chromatogram of the ICPMS on the selenium. Then we get the total ion chromatogram on the electros-PMS. We have the extract ion chromatogram of the selenium peptide, and then if you zoom in that peak here, we see the nice selenium pattern, distribution pattern on the mass here. So we can actually assign the identity of the peak to one of our selenium peptides. We did get a little bit further as well. So we did HPLC Isotope MS experiment to get actually the accurate mass on the natural and the alkylated on the natural and the enriched peptides. We use different setup. In this case, water caseating column with acetonitrile formic acid gradient, low flow rate, and you can see actually here the experimental, the theoretical mass, and we get a relatively small error for our natural peptides. And we did the same to our enriched peptides again, so quite a relatively small error. So we've got the accurate mass, so we can actually for sure say we're looking at the right species. Then we had to optimize the triptych digestion, the peptide alkylation and extraction procedure. As you can see here, for example, if you add the alkylated spike peptides to the serum mixture before any other, you can see here the transformation of the spikes, which we don't know what is, and we want to avoid it. So that's not very good. And again, as well, if you add the unprotected peptide spikes to the serum sample mixture, again, we see some unknown transformation of the spike, and we don't want that because we want the spike and the natural peptide to be the same. Finally, we optimized our digestion alkylation procedure. So both the peptides in the serum, reduction, followed by the digestion, protein precipitation, again reduction, centrifugation, alkylation, lyophilization, and then we could directly inject them into the ICP-HPLC MS. So that was actually that's our optimized HPLC method, as you can see here. That's all the ICPMS tray. So that's our HPL setup. So we've got a methanol and a methanol water gradient with formic acid, slow gradient up here, 25 microliter injection. That's our EE peptide and our ENL peptide, and you see here, so in 76 it's a little bit enriched because they are spiked, so that's what you actually want. So, and it's quite a nice baseline separation of other selenium species, so that that's what we exactly want, so we can actually use to quantify, and our IDMS approach is actually valid. A little bit on the ICPMS side, detection side, we use the new 8800 drill quad system, so we can measure in mass shift mode, selenium 76 and 92, 92 and 878 and 94, which is the optimal removal of interferences such as argon-based interference, we had to set up in the organic mode. We need a plug-in skimmer and sampler, now a torch, spade chamber, and additional gas supply. Looking at the IDMS determination of selenium protein pain, so we did so that's the results here. So on the ENL peptide and the AL peptide, so it's got a uncertainties quite low and expanded uncertainty, and they both agree with the measurement uncertainty. We got no lung contribution. The 4 values agree with the uncertainties. We got some spike recovery experiments. Some other group did the confirmation experiment, and they did get 60 with a different techniques, so they get 63.9 microgram per kilogram. We get 60.6 microgram. We also used the QC, the BCR, which we have 56.2 and 58.3, and this is a good agreed value with the literature. We also were able to create an uncertainty budget. So, and as you can see here, the most of the contribution is the measurement of the isotope ratios in both the blends, and of course the uncertainty associated with the primary standard. Coming to the conclusion, so hopefully showed you that we had a systematic approach for the accurate determination of selenium protein via the selenopeptides using species-specific isotope dilution, and the method has been developed and validated. With this approach, we are enabled to quantify selenium protein P at relevant clinical levels down to 60 microgram per kilogram with a relative expanded uncertainty of 5%. The method offers a potential for the use of as a quality control of measurements in the clinic, and furthermore, it can help also for the provision of reference materials for clinical trials and certification of reference materials. Looking further ahead, we still have problems with low sensitivity because of the low concentration. So hopefully, by increasing the sensitivity, we're able to get better isotope ratio precision, so we can use different cell gases. Maybe looking at different selenoproteins in plasma, so glutathione peroxidase maybe is another one. And then, of course, that's ambitious, but of course, we're trying to even look at lower levels, for example, in microsection of tissues and brain. Next year, there's going to be an intercomparison lab study organized to look at selenoprotein P in serum and various NMIs around the world to participate in that. So that's going to be quite interesting. And of course, one big aim is because selenium proteins is limited, but if we're looking at sulfur, so sulfur is available in almost every protein. So if we can extend those approach to the detection of a sulfur enriched peptides, that would be, of course, quite a good thing. If anybody of you in the audience is interested in any further reads, we've got that's been published, our work in analytical chemistry. So feel free to have a look, and if you're interested, or you can ask any questions later on. Leaves me with final thank yous. Thank you to my co-authors. Thank you to my colleagues at the NML, the inorganic team predominantly, and Heidi as the team leader. Thank you to my other colleagues, and thank you to our funding, Euromed, and well, of course, thank you very much for taking the time out today and listening to our presentations. Very much appreciated. So thank you very much.
Thank you, Christian. Really, really, really lovely presentation. So much hard work in all of that. I'm going to introduce our next speaker now, and that is Nancy Wanner. Let's bring Nancy's picture up. Hi, Nancy. Hi there. Our third speaker today is Dr. Nancy Wanner from, is it SCKCEN?
That's right.
Fantastic. Nancy's PhD project was entitled The Method Development for the Characterization of Spent Nuclear Fuel Using Isotope Dilution, HPLC ICPMS, and was performed at the Belgian Nuclear Research Centre in collaboration with Ghent University. Nancy successfully defended her PhD in March 2021, and since then she's been working on the radiochemistry group at SCK-CEN to develop new analytical methods. Nancy will talk to us today about the quantification of uranium, plutonium, neodymium, and gadolinium for the characterization of spent nuclear fuel using isotope dilution, HPLC SF ICPMS. And I will just swap the presenter over, and we'll be okay. You want to just excellent. I think that all looks good, Nancy. I will leave you to it. Thank you very much.
All right. Thank you very much for the introduction and for this kind opportunity to present the work done in my PhD, which was to develop a method to quantify uranium, plutonium, neodymium, and gadolinium for spent nuclear fuel characterization using isotope dilution with HPIC coupled to a sector field ICPMS. Let me first introduce to you what happens in a nuclear reactor. Heavy, unstable nuclides are bombarded with neutrons, and they undergo fission, which means that they split into two lighter nuclides, which are more stable. This process creates energy in the form of heat and radiation. So what are we interested in knowing? We would like to determine the burnup of the fuel, and that gives us how much fission underwent the fuel in the reactor. And this burnup is determined in % fissions per initial metal atoms. Burnup is calculated using a fission product monitor, such as neodymium-148, and the initial components, and the main components of the fuel. In the case of experimental fuels, and those are fuels which have burnable poison added to them or a neutron absorber, such as gadolinium, and that halts the fission chain reaction. There are different ways to characterize spent nuclear fuel, which we are also doing in this, I also did in this PhD. So one of the ways that I looked into is by characterizing the fuel using destructive analysis methods. And these are done in specialized laboratories where the fuel is dissolved inside hot cells and is analyzed using either alpha spectrometry, thermal ionization mass spectrometry, or inductively coupled plasma mass spectrometry. These techniques are hindered by interferences between the fuel components, and that is why it is necessary to separate the fuel components prior to their analysis. This can be done using offline gravitational chromatography. However, this method is time-consuming as it can take up to 2 weeks to separate the lanthanides, for example. And it also puts a risk of radiation on the operator. Instead of using gravitational chromatography, one can use high-pressure ion chromatography. This is a faster method, and it also lowers the dose rate which the operator comes in contact with. Additionally, HPIC can be coupled with ICPMS. However, no physical connection can be done with the column, with the back end of the column, and alpha spectrometry or thermal ionization mass spectrometry due to the operating principles of these two instruments. So my goal was to develop an accurate and fast method to determine the mass fractions of the elemental as well as the nuclide-specific compositions of uranium, plutonium, neodymium, and gadolinium in spent nuclear fuel. This was done in several steps. The first one was to separate or develop a method to separate the elements of interest, and that was done by developing an dilution program on the HPIC. The second step was to investigate different instrumental parameters and different calculation methods to obtain the most precise isotope ratios in the transient signal which we were dealing with in this case. And lastly, we used isotope dilution to determine the elemental mass fractions of our elements of interest in 2 types of spent nuclear fuel. And we compared our results with those of an accredited method which is currently used at SCKC, and that is using offline chromatography with alpha spectrometry and thermal ionization mass spectrometry. But we will look at that in a second. First, I would like to show you a short video of the instrumental setup used for this work. I will just mute the volume so you can hear me. So here you see the sector field ICPMS. It's an element 2 by Thermo Fisher, which was used. And the inlet system is placed inside of a glove box for handling radioactive material. On this side, you see the HPIC pumps. So the first pump takes up the eluent to the column, which is placed inside the glove box. And the second pump takes up the internal standard to be admixed with the column effluent. These are 2 quaternary gradient pumps, which you see here. The column is placed inside of the glove box, and the one used for this work is a mixed bed ion exchange column. It's a CS5A by Biogenic. So in a run, the autosampler takes up the sample from the vial, as you see here, and through a peristaltic pump into the first of the 3 Rayodyne valves, which is equipped with an injection loop, which does the injection of the sample onto the column. After going through the column, the sample goes to the second Rayodyne valve, which directs the flow to either mass spectrometry or UV-Vis. And if we choose to go to mass spectrometry, the flow goes to the third valve, which takes it up to the inlet system of the ICPMS. And here you see the nebulizer and the spray chamber. We used 2 software, Chameleon for the HPIC and the element 2 by Thermo for the ICPMS. So let me share with you now results obtained with this setup. You see here an example of a chromatogram obtained from the injection of a spent fuel sample onto the HPIC. And you see all the elements of interest are nicely separated. So plutonium and uranium were eluted with nitric acid, whereas the lanthanides were eluted with a gradient of 0.1 to 0.15 molar oxalic acid at a pH of 4.5. So the elements of interest are separated with a resolution greater than 1, and they each elute with a single retention time, which is suitable for isotope ratio measurements. I would like here to introduce the 2 types of peaks that came along the way. So what you see here are chromatographic peaks of neodymium nuclides, and they are made of sequentially obtained data points. Each of these data points comes from a spectral peak, which is the flat top peak, which is characteristic of the sector field ICPMS. So we investigated 2 different dwell times, and those affected the number of data points on the chromatographic peaks. And we also investigated different mass windows, which affected the number of lines which you see in the spectral peaks, or which we call samples per peak. So we kept 20 samples per peak, and we chose different mass windows in the spectral peak. So with a mass window of 2%, we would be acquiring 1 sample per peak, whereas with a mass window of 150%, we would be acquiring the full 20 samples. We also calculated the isotope ratios in the transient signals using 3 different methods, and those are the widely used in the literature, the most widely used in the literature. The first one is point by point, in which the intensity of one isotope is divided versus the intensity of the second isotope, and the average of the intensities is taken as the isotope ratio of interest. The second method of calculating isotope ratios is linear regression slope, in which the intensity of one isotope is plotted versus that of a second isotope, and the slope of the linear regression of the data is then taken as the isotope ratio. And the third method of calculating isotope ratios is using peak area integration, where we are simply dividing the area under the chromatographic peaks of two different isotopes. So let's first look at the effect of the dwell time on the precision of isotope ratios. So on these two graphs, you see that with the shorter dwell time of 10 milliseconds, we have more samples per peak, per chromatographic peak, compared to those obtained with a dwell time of 30 milliseconds. And here you see the isotope ratio of neodymium-142 to neodymium-146 obtained using the 3 calculation methods and the 2 different dwell times. And as you see here, peak area integration gives us the most precise isotope ratios and is not affected by the dwell time. The second set of results shows us the improved precision of isotope ratios with the different mass windows and the different calculation methods. So the precision of the calculation methods were compared to the theoretical precision that is obtained using counting statistics, and with a mass window of 25%, a peak area integration, we obtained the closest precision to the theoretical one. We also determined the repeatability of our 3 calculation methods. So here you see an example of neodymium-142 to 146 again, with 10 different injections of the same standard. And you see here that with peak area integration, we obtained the least spread, which gives us the best repeatability. And this was obtained with a mass window of 25% and with a dwell time of 10 milliseconds. So the third step, now that we have optimized the instrumental parameters and chosen to calculate the isotope ratios with peak area integration, we used these in the third step, that is to characterize 2 different spent nuclear fuel samples using isotope dilution. So in this graph, you see the principle of isotope dilution, where a sample and a spike of totally different isotopic compositions are mixed together to form a blend, and the blend is then measured using mass spectrometry. We use the following equation for isotope dilution to determine the mass fraction of the elements of interest in the samples. So in which the mass was obtained from the balances. We took also the spike information from the certificate of the certified reference materials used to spike the sample, and the rest of the elements in the equation were actually measured or based on the measurements of the sector field ICPMS. So here we are comparing the elemental mass fractions which were obtained for uranium, plutonium, neodymium, and gadolinium using our method, with compared with the accredited TIMS and alpha spectrometry method. And you see in these 2 graphs for both types of fuels, there is an agreement between the different elemental mass fractions obtained with the 2 methods. We could also determine the nuclide-specific mass fractions. Here you see an example of the gadolinium nuclide mass fractions in a gadolinium fuel, or a fuel that is spiked, that is in which gadolinium has been added as a neutron absorber. And you see that there's a good agreement between the results which we obtained using the HPIC, ICPMS, and compared to those obtained using TIMS and alpha spectrometry. However, you see on the left-hand side of the table that the precision obtained using TIMS and alpha spectrometry is lower compared to that obtained using our instruments. So in conclusion, we developed a new method to characterize spent nuclear fuel safely, precisely, accurately, and rapidly using isotope dilution, HPIC, SF, ICPMS. This method, compared to the currently applied one of TIMS and alpha spectrometry, is safer for the operator and increases the sample throughput. Seeing that the isotopic mass fractions were in agreement with the two different methods, but they were more precise with TIMS and alpha spectrometry, and taken into account the time savings, obtained with this method, we can still use this method to determine the amount of sample to be spiked for isotope dilution, TIMS, and alpha spectrometry. I would like to thank both sides which collaborated to make this work happen. So from SCKCEN, my mentor, Karen Van Hoeke, Andrew Dobny, and Mirella Vasile, and my supervisor from Ghent University, Frank Van Hake, and again, from both sides, all the people and the colleagues who helped and supported me during this work. I would like to thank you all for your attention. And if you have any questions or comments, please feel free to ask in the questions and answers section of this webinar. And if you don't get to stay until then, please feel free to contact me via email. Thank you.
Thank you, Nancy. That was really clever. And you're our first video on these seminars. So well done for doing that. That was really nice. Yeah, really, really nice. Okay, thank you. So if we move to our last speaker for the session, we do. Okay, so our last speaker for the session is Elliot Hamilton. He's an environmental chemist working within the inorganic geochemistry laboratories at the Centre for Environmental Geochemistry at British Geological Survey. His primary research areas interests are the development of speciation methods for the measurement of potentially harmful elements in environmental and biological matrices, and assessing the availability and fractionation of pollutants in contaminated soils using microspatial sampling techniques. Elliot's talk today is entitled Separation Anxiety, A Personal History of Chromatography for Chromium Speciation. And as Nancy reminded you all, can I just remind you all, if you'd like to ask any questions, to put them in the questions drop-down box or in the chat, and we can discuss those after Elliot's talk. Thank you, Elliot. I'll pass over to you.
Thank you very much for the introduction, Jackie, and thank you to all of the presenters. Been some really interesting work that's been sort of presented over the course of this session. So my name's Elliot Hamilton, and today I'm going to be going over how chromatographic separation techniques were used for my part-time PhD, which I completed back in July last year. So the title of that, of the sort of thesis, was Novel Methods for the Assessment of Chromium Species Bioavailability. And I'll be sort of going over, as sort of case studies, the three main experimental chapters that were published. So to start off with, why we should be interested in chromium. So thanks to Christian for sort of giving a good succinct definition of elemental speciation for me. But so it's present in the environment as two different chemical forms or species. So the trivalent form is an essential micronutrient for glucose and lipid metabolism, which has led to its widespread use as a putative weight loss supplement. Whereas chromium-6, hexavalent chromium, is toxic and a recognized carcinogen through inhalation, with a UK residential category 4 screening limit of 21 milligram per kilogram in soil. So the majority of chromium-6 is released into the environment through industrial processes such as chrome plating and leather tanning. And due to the negative charge of its compounds, chromium-6 is more bioavailable and more mobile in soil water systems than chromium-3. And is therefore more likely to be taken up into plants, although this may not be the form that it actually remains in. So the bulk of this presentation will now be a series of case studies covering the experimental chapters of my PhD, the first of which concerned the determination of chromium-6 in soils collected from the Copperbelt province in Zambia, and the use of a single spike speciated isotope dilution technique. So why Zambia? So there are two reasons. So my PhD started at the same time as a Zambian student, Belinda Kaninga, as part of a Royal Society Department for International Development capacity strengthening initiative. And the two projects required reconnaissance work to site future crop trials and also understand the elemental spatial variation at a number of prospective sites. Also, the Copperbelt province in Zambia has been extensively mined over the last century, as the name would suggest, for copper, but also for cobalt. So there's been wide-scale anthropogenic contamination of water and agricultural soil, but little attention has actually been placed on the potential for chromium contamination, and particularly its species. So a number of sites were surveyed, with the most work undertaken at this site to the south of the town of Kalalushi. Here's a UAV photo just showing the sort of corner of that very large tailings dump, with this village called Mugala village, situated in very close proximity. Quickly, a nested sampling design was implemented at the site to understand the spatial variation of elements and additional soil parameters such as pH and loss on ignition, which was then enabled us to interpolate a surface to understand where there were areas of hotspots and potential dispersion of tailings material. And then follow-up sampling was then undertaken at the time for potential glasshouse trials, with the need to then determine the chromium-6 that may be present within the soils. So the speciated isotope dilution was modified from this reference. So as we've heard in the previous talks, there's a real need to ensure sort of full equilibration of the spike and sample. The main difference for speciated isotope dilution is that the isotopically enriched spike is in the same speciated form. So there's an isotopically enriched spike of chromium-6 that's being added to the sample. The equilibration is achieved through microwave-assisted extraction with EDTA, which is acting to help to reduce the likelihood of any chromium-3 oxidation to chromium-6, because chromium-3 will form the chelated EDTA form. And then following a microwave-assisted equilibration, the sample can then be centrifuged and supernated and collected for HPLC ICPMS analysis. So I used a 8900 triple quad, although not in a particularly sort of inventive mode, just in simple collision mode with helium, to reduce the likelihood of or the potential bias from argon carbide interference. and then a bioinert HPLC system was used to reduce the issue of stainless steel systems causing chromium contamination. And then with an anion exchange column, so with the chromium-3 EDTA complex, we're able to resolve both species on an anion exchange column. We're able to achieve an isocratic separation of both species within 7.5 minutes. And the NIST certified reference material hexavalent chromium in contaminated soil, the low level was processed in the same way as the samples, so subjected to the single spike isotope dilution, and gave a good average recovery across 2 separate digestion batches. So very quickly, as we with respect to the results, as we would expect for the very low pH, quite highly weathered soils in that area, the chromium-6 was below the UK screening level. There's no Zambian actionable limit for chromium-6, which suggests, although of course no exposure assessment was actually undertaken, that there's a low risk for chromium-6 at that site. The primary impact for this work is that the reconnaissance work, the interpolated plots produced for that were then subsequently used by Belinda to cite crop trials, which were concerned with investigating how different agricultural management practices may impact on the transfer of heavy metals into staple crops. So the second experimental chapter, the shift of focus was then more on the sort of species transfer mechanisms that plants that would be grown at the study site may actually experience. So an experiment was then devised to actually understand how a staple crop such as spinach, primarily chosen for its ease of handling, how that may actually, or what sort of processes it may undergo to actually try and reduce the likelihood of chromium-6 toxicity. And this was done using isotopically enriched forms of chromium-3 and 6. So the current understanding for species transfer mechanisms into root cells is that the trivalent form is transferred passively, whereas chromium-6 is an active transfer process which does expend energy on the part of the plant. And then in the presence of an anion such as sulfate, the sulfate will then compete with and will be preferentially transferred through the active transfer mechanism. So really an experiment needed to be devised to start to deconvolve the following issues. So do plant roots actually cause any conversion of chromium species in the low pH environment of the rhizosphere, which exudes organic acids? Does that contribute to chromium-6 reduction? What is the dominant species of chromium following transfer into the root cell, and does sulfate compete with or promote transfer, as was suggested in literature at the time that this experiment was devised. So using a design adapted from Humphrey et al., spinach plants were germinated and grown in silver sand, just to reduce the likelihood of a chromium background, although of course this was also balanced by the use of isotopically enriched treatment solutions. And then after approximately 21 days, when they'd reached maturity, the plants were harvested and then subjected to a range of chromium-3 and 6 concentrations, and also a fixed chromium-6 concentration with differing concentrations of sulfate. We really want to start to understand, during uptake, does the plant have a exclusive transfer of one or more species? Do we have conversion following transport, or for example, do we have conversion prior to transport in the low pH environment of the rhizosphere? So following on from the treatment, so this was just a short-term exposure. After 24 hours, the root tips were then removed from the plants, and then the solutions corresponding at the time of the treatment to the active and passive pathways were then extracted, and the same HPLC ICPMS setup as seen in the soil chapter was then employed. And, don't worry, we have reduced the size or the length of that tubing. So the results themselves are quite complex to summarize in a a succinct way for the scope of this presentation. So I'm just going to focus on the impact of the sulfate. So the treatment solutions with a fixed concentration of 500 ppb of 53 chromium 6. Excuse me. And we have the passive and active solutions, the chromium concentration measured by HPLC ICPMS, and then we have either a control with respect to sulfate and increasing sulfate concentration 150 and 250 milligram per litre. A 50 was attempted but didn't yield enough solution. So probably the most, and the species blue representing chromium 3 and red representing chromium 6. So the most immediate thing to note is that chromium 3 is actually the dominant species in both the passive and active solutions, which suggests that there has been a reduction of the chromium 6 that was added and was significant over the reduction that just naturally occurred in the solutions where there were no plants. So this, but whether this reduction process actually occurred prior to or following transfer can't be differentiated with this experimental design. There's a visual trend of an increase in the reduced chromium 6 through the active mechanism with increasing sulfate, although this isn't supported by non-parametric testing. However, there is a significant difference between the passive and its respective active means, which does imply a promotion effect for sulfate through the active mechanism, which is supported by recent literature. However, it is important to note that plants are very likely to and are capable of changing their transport mechanisms depending on the external demand. And so therefore, this is simply a snapshot in time of what a plant may experience in the presence of chromium 6. And so more work is obviously needed to understand if this promotion effect is universal, and also what the implications are for actually managing the potential risk of transfer of chromium 6 into plants. So the final experimental chapter could be considered as a a consolidation of the previous chapters, and was a proof of concept to investigate the potential for microdialysis coupled to HPLC ICPMS to continuously sample and measure chromium 6 in soil solution. So microdialysis is a passive sampling technique which was originally developed for neuroscience and pharmacokinetic studies to investigate neurotransmitter release in interstitial fluid. But it's being increasingly used in the field of soil science to allow in situ sampling of solutes without disturbing the environment being sampled. So microdialysis works on the principle that you have a small probe, typically 0.5 mil in diameter, and that's implanted into soil or a solution, and a perfusate solution is then pumped into the probe, which then subsequently flows over the semi-permeable membrane contained at the end of the probe. This then establishes a diffusion gradient between the sampled medium and the perfusate, and then causes solutes to diffuse across the membrane, with the resulting solution that's pumped out of the probe containing the sampled solutes for either offline or online analysis. So the main benefits of microdialysis for soil sampling are the much higher spatial and temporal resolution compared to existing passive sampling techniques such as diffusive gradients in thin films, DGTs, or Rhizon samplers. So this is able to sample at much finer scales than these techniques, which are more relevant to what a root would be experiencing, or the sorts of interactions that may be occurring at sort of the rhizosphere. And the high temporal resolution means that you can investigate the turnover of solutes due to its capability to continuously sample, which means you can understand much better the timescales for the very fast soil chemical reactions that currently no technique is able to achieve. The primary limitations, though, are associated with the efficiency of sampling, which is due to the multiple resistances that could be present that impact on the capability of the solute to dissolve towards the probe, such as pore size, the flow rate through which you sort of deliver the perfusate into the pump. And these are all considerations that still require some sort of reconciliation before the technique can be more widely adopted. So this is a picture of the setup for the MD HPLC ICPMS technique. So the syringe pump was delivering a sort of a millicu perfusate into a sort of a moistened soil sample. The HPLC setup was again the same as what was seen in the soil and the root solution work. So an isocratic setup, just simply that it was all connected to a manual sort of a Reodyne flow injection valve equipped with a 20 microliter loop. The system was able to then sample at 15-minute intervals the concentration of chromium-6 sampled within the soil. That was the time taken for fresh solution to fill the dead volume of the dialysate tubing and double fill the loop to ensure the best precision. And then the valve was manually switched, and then the samples injected into the anion exchange column, and the ICP triple quad was then manually sort of triggered to start the acquisition. So as I said at the start of this sort of case study, the results are more a proof of concept at this stage. There's certainly an intention to try and reduce the sampling resolution or sampling density even further, but the purpose of this proof of concept, 3 separate soils were sampled just to understand what the potential limitations of the technique may be. So we had 2 sieved soils, one that was anthropogenically contaminated and one that had a sort of a geogenic total chromium concentration over 600 milligram per kilogram. And then this milled contaminated soil, which was the soil reference material used in the soil chromium-6 determinations. You can see that in all cases that the technique is able to offer a fast and precise fine scale measurement of chromium-6, with the main sort of limitation being this milled soil, a much finer particle size that then subsequently creates this variation even with the lower chromium-6 soil. We're able to fairly reproducibly sample before it then goes beyond the sort of detection limit of the system. And we're even potentially sampling a more immediate labile pool at the start of this, and then sampling a more sort of consistent pool, although of course the resolution is still not sufficient to be able to completely confirm this. So there's really the potential for this, as shown by these results, is that the MD HPLC ICPMS technique could offer much higher resolution data to better inform geochemical modelling and the assessment of rapidly, or of sort of a rapid turnover of sort of micronutrients or potentially harmful elements. With simple modifications to the chromatographic setup would then enable the speciation of other elements such as iodine or selenium, which are critical for sort of food security issues. So I would like to extend thanks to the following people who were instrumental in my progress over the course of my PhD, and thank you to all of you for listening to this talk, and take any questions.
Hi everyone. So I'm just going to bring everybody back on, if that's okay, and we've got some questions that we'll just go through. So I keep pressing the unmute button and it just doesn't work. We'll try it again in a sec. Let's bring the cameras on. That's not, there we go. Hi Christian. Let's see, for Roberto. Brilliant. Everybody's there. That's really good. Okay, there's not too many questions. Thank you all for your presentations. Just trying to unmute you all again. No. You'll be all pleased to hear that the next presentations that we've organised will be on Zoom. Oh, there we go. It's playing now. Christian, you've got sound. Nancy, you've got sound.
Yes.
Roberto, just waiting on you going from orange. There we go. Well done, everyone. Sorry, it makes it look like I can't do it, but I keep pressing it and it doesn't work. So thank you all for your talks. Amazing. And to say that Elliot, Nancy and Roberto, that you're recent PhD graduates, seriously, your projects blew my mind. It was so impressive. That's not to say that, Christian, you're not impressive, but just the fact that these guys have just graduated in the last year or so, I mean, it's just phenomenal. So I'd like to thank you all for your great presentations. We have a couple of questions, and the first one is to Roberto. Roberto, thanks for the insight into using HPLC for nanoanalysis. Should my feet be worried about ionic silver and silver nanoparticles leaching from socks? Are the levels you presented from the washing enough to be concerned about in the environment?
It's a good question. I don't know really how much concentration these nanoparticles can be toxic for humans. We have only worked on the method development of the HPLC ICPMS method, and we can detect 2 fractions of different sizes of silver nanoparticles in the socks, and also ionic silver, but I cannot tell you if the levels that we found are toxic for humans.
Okay, thank you for that. Nancy, I have a question for you. Thank you for a really clear presentation of your PhD work. The uncertainty difference between your method and the alpha TIMS is certainly balanced by the time saving and the safety. How important is the uncertainty or the precision to the final measurements?
Taking into account the nature of the samples and the reason why we are doing this work, I would say the accuracy and the precision is actually plays an important role in choosing the correct method to be used for the characterization of the sample, of the nuclear sample at hand. So that is why, in terms of accuracy, HPIC sector field ICPMS, the method that I developed, was less accurate than the TIMS and the alpha spectrometry accredited methods. However, it can still be useful in terms of determining the concentration of the sample the concentration of the elements that I investigated, so uranium, plutonium, neodymium, and gadolinium, in the samples to be spiked for isotope dilution, TIMS, and alpha spectrometry.
Okay,
well, does that answer the question?
It does, and you know, I'm sure the world of nuclear demands a greater amount of precision than the world of biological, which is where I come from. but your precision still looked pretty good. I appreciate different to Tim's, but if you take the safety into account. Elliot, I have a question for you. Do you think the size of soil particles generated by milling influences your chromium levels? If so, could you have controlled the sizes from your milling device?
The CRM itself was purchased separately, so that was the NIST CRM. But yes, I would expect the finer particle or there to be a higher chromium-6 concentration, and that certainly would be built into more of an investigation into, specific particle size effects, maybe trying to replicate, sandy, silty, loamy soils to really understand and probably do a more sort of holistic approach to the microdialysis sampling to understand the artifacts that may be present.
Okay. Oh, no, that's a good point. Roberto, I just wondered, it is just a question from me. So you mentioned that this selenium nanoparticles were 4 to 6 nanometers, and I just wondered how low you think we can go. Nanoparticles and single particles with ICPMS. I mean, that's really, I mean, we've used 5 nanometers silver, and that's as far as I've gone, but 4 to 6 for selenium with its different ionization, that just seems amazing.
Can you repeat the question? Okay.
Sure. How small nanoparticle do you think we can go? Can we go lower than 4 nanometers for selenium?
By HPLC or by single particle?
Any just what you think, if you think we can go.
By single particle, our detection limit was around 20 nanometers.
Okay.
Our size detection limit. So we decided to use the HPLC ICPMS approach to investigate the presence of smaller particles in the yeast cells. So the limitation for this application is the resolution between the smaller particles and the ionic low molecular selenium species. So we can distinguish between these particles of 4 nanometers, and we also work with, for example, iron nanoparticles of 3 to 5 nanometers, and we can distinguish them from the ionic species. But we are in the limit, I think.
I know. I just wonder how long it will be before we're getting to what's the next one? Phenton particles. How low will it go? So just that closes our talks today, everybody. Thank you so much for joining. I've had 3 comments saying wonderful talks to you all. And I mean, it set the bar really high for a chromatography session. I wasn't quite sure how it was going to go, and you've all delivered amazingly well. Brilliant. I just wanted to thank all the speakers and everyone in the audience attending, and to let you know that the next virtual session, D is for Development, will take place on the 28th of July at 2 o'clock in the afternoon. We're still looking for another speaker for that, so if anyone would like to volunteer themselves, that would be amazing. And feel free to comment, to email me any comments at any time. It's jackie.morton@hse.gov.uk. So that's it, everyone. Thank you so much.
Thank you very much.
Thank you for having us.
Thank you much,
Okay. Bye.
Bye.